Now moving to the next page, talking about spleen. Spleen is located in left upper quadrant of abdomen, anterolaterals to the left kidney, protected by 9th to 11th ribs. Now splenic dysfunctions, or we can say post splenectomy, we will talk it about later, and let us talk first about the structures of a spleen. So it is covered with the capsules, and inner invaginations is known as trabecula, as in lymph node. So consist two parts, that is the red pulp and white pulps. Inside we can see the white pulp, and the outermost part is red pulp. So red pulp consists sinusoids and reticular fibrous framework. Reticular fibrous framework. Similarly, for white pulp, we can see follicles. Then follicles part is like mental zones and germinal centers. Similarly, the marginal zones and periarteriolar lymphoid sheath, that consists T cells. So here we can see that is periarteriolar lymphatic sheath, that is a part of white pulp, contains T cells located within white pulps. That is periarteriolar lymphatic sheath. Periarteriolar lymphatic sheath contains T cells located within white pulp. Similarly, follicles contains B cells located within white pulps. Similarly, the marginal zone contains macrophage and specialized B cells. Macrophage and specialized B cells, site where antigen presenting cells, that is APC, antigen presenting cells captures blood-borne antigens for recognitions by lymphocytes located between red pulp and white pulps. So marginal zones is in between, located between red pulps and white pulps. Now let us discuss this. So we are talking about splenic dysfunctions. Example, that is post splenectomy, seen in sickle cell disease, that is autosplenectomy, in which the decreased of IgM or complement activations, decreased C3b opsonizations, susceptibility to encapsulated organisms. Now post-splenectomy findings are John Howell-Jolly bodies, that is nuclear remnants, target cells, thrombocytosis, loss of sequestrations and removals, lymphocytosis, that is loss of sequestrations. Now vaccinate patients undergoing splenectomy or with splenic dysfunctions against encapsulated organisms, that is pneumococci, HIV, meningococci. Now talking about thymus, located in the anterosuperior mediastinum, site of T cells differentiations and maturations. Encapsulated, that is thymus. In thymus epithelium is derived from third pharyngeal pouch endoderms, whereas thymic lymphocytes are of mesodermals in origins. So cortex is dense with immature T cells. Cortex medulla is pale with mature T cells, and Hazel's corpuscles containing epithelial reticular cells. Adult corpuscles containing epithelial reticular cells. Normal neonatal thymus is cell-shaped on chest X-ray. Asterix involves by age 3 years. Here we have T cells, that is thymus maturations, B cells, that is bone marrow. Absence thymic shadow or hypoplastic thymus is seen in immunodeficiencies. Example: severe combined immunodeficiency and Dyer's syndromes. Thymoma is a neoplasm of thymus associated with Myasthenia gravis, superior vena cava syndromes, pure red cells aplasia, good syndromes. This much for now.